right, good morning, everybody. Let's announcement. So the handouts here, both the PowerPoint printout and a hard copy of the problem set that's due on Sunday. The Word file is up on Moodle, so you can either work on this piece of paper that's handed out, or you can work on the Word file that you download from Moodle. Either way is fine. Remember that the problem set is open book, open notes, open friends, open internet, except that, well, if I find out that you basically posted the question on the internet and then asked somebody to answer it for you, I won't be pleased with that. But you're certainly allowed to look up things on the internet. What else? Any questions about the problem set? It is warm in here. I don't know how it's really stuck. I'll make some mess. Any questions about the problem set due on Sunday? So the hard copy, even if you work on your own Word file, the hard copy is due at the CST desk at 11:30 p.m. on Sunday. Also on Moodle is a copy of the lab protocol for Thursday. I'll print out copies for in lab. I probably won't print out a copy for everybody, but you can print out your own copy if you want it from Moodle. So the PDF of Thursday's lab is available on Moodle now, and I guess I would request that you skim it. You don't have to study it, but that you skim it to prepare for what's going to come up in lab on Thursday. We're going to be dissecting out the human heart, and we will be using pig hearts to enrich the human heart experience. So we'll start with cardiac anatomy on Thursday. Today we're going to continue this whirlwind tour of the key concepts from cell physiology. So last week we talked about ions and primary and secondary active transport and some of the transport mechanisms. We'll do a little bit more of that right now, and then turn our attention to some really important concepts: receptors, second messengers, signaling pathways. These are things that many of you had in great detail in A&P1. All of you had in some detail in cell physiology. It's going to be really important for cardiac physiology because the heart is both it has receptors that modulate its strength and rate of beating. It is a muscle, so it uses the mechanisms of muscle contraction that when we focused on skeletal muscle, we will take some of those principles and apply them to cardiac muscle. There's some significant differences, but the principles remain the same. So all of these things, messengers, signaling pathways, electrophysiology of membrane potentials, action potentials, and synaptic transmission, molecular mechanisms of muscle contraction, they're all going to become very important very soon in the cardiac section, cardiovascular section. So here's a figure from your textbook that highlights some of the transport. So going back to transport. So let's see. The sodium-potassium ATPase is essential, right? So this one here, that's the sodium pump. Pumps sodium out and pumps potassium in. And we'll see the importance of that over and over again in most of the sections of this course. The calcium pump. This is the calcium ATPase that is especially important in nerves, which we won't focus on, but muscle, which we will focus on. Remember, there are two types. There's the circa that pumps calcium into the sarcoplasmic reticulum or the endoplasmic reticulum, and there is the plasma membrane calcium ATPase that pumps calcium out of the cytoplasm into the extracellular space. Both of those are important for keeping resting calcium concentration in the cytosol very, very low. Let's see, in this course, we will talk about this. This one is going to be important when we get to the digestive tract. when we get to digestion, this sodium amino acid co-transporter, secondary active transport, is going to be very important. But that's going to come towards the end of the course. The sodium proton exchanger, we probably will skip over. We won't focus on that one. The sodium calcium exchanger is going to come up important actually today and in the problem set because it's important in cardiac and cardiovascular. Remember we talked about another mechanism for pumping calcium out of the cytosol, a sodium calcium exchange transporter, secondary active transporter. This one is going to become very important in respiratory, which comes after cardiovascular. Facilitated diffusion of glucose is going to be in the digestive. Calcium channels, potassium channels, and sodium channels are going to be important right now. So this figure from your textbook, which highlights the basic transport systems that we're going to be using in this course, is important throughout the course. So let's do a clicker. Hopefully you all remember to bring your clicker. I didn't bring extra copies today. I'm going to see how compliant you are with regard to bringing your clickers. If you have already lost your clicker, please let me know and I can give you another one. But I asked you to bring your clicker. All right, so if the sodium potassium ATPase is inhibited by 50%, that's a huge inhibition, what will happen to the intracellular concentration of the following ions? And remember to consider secondary active transporters. So if the sodium potassium ATPase is inhibited, what happens to intracellular sodium concentration? Show me your answer. Okay, I'm seeing a lot of the correct answers, which is A. If the pump is inhibited 50%, then the cell is less efficient in pumping sodium out, and all the other leakages and channels and transporters will happen, and the sodium concentration would rise because one of the most important ways of getting it out of the cell has been inhibited. It's as simple as that. So the main concept here is to be thinking about leaks and channels when you try to apply the whole system. Inhibiting one piece and then remembering that sodium will leak in down its concentration gradient through channels and just leaks would allow the sodium concentration to increase. Okay, so that's pretty straightforward. Okay, what will happen to intracellular potassium? Show me your answer. Yeah, by the same logic, the potassium concentration inside would decrease because the thing that is pumping it in, the potassium that is high on the inside would leak out. And the potassium concentration on the inside would decrease. Now there could be some compensatory changes. So, but the sodium potassium ATPase is so important for the homeostasis of sodium and potassium that both of these will happen. Okay, so now what would happen to intracellular calcium? And I'm just going to skip to the answer because that's a homework problem. That's part of the problem set. So we're not going to give you the answer here, but we'll talk about it if necessary, depending on the answer that you write for one of your homework questions. It's related to question 4. Okay, and what about glucose? what would happen to intracellular glucose if we inhibit the sodium potassium ATPase? Here. It's reflected in your color scheme of the class right now. So I think that the best answer, I think, is D. Could go either way. So let me ask you to work together, because many of you put C, no change, which is kind of, okay, if it could go either way, maybe it doesn't change. I think it could change, but it could go either way. And you could make an argument for how the concentration of glucose inside the cell would increase, and you can make an argument for how the glucose could decrease. So I'm going to give you like 2 minutes to talk to each other and try to come up with an argument for how glucose could increase and come up with an argument for how glucose could decrease. That is to say, why it could go either way. Okay? So let me give you a minute to do that. Okay, does someone want to volunteer? Does someone want to volunteer to answer either of these questions? An argument for either way, your choice. Clock.
So I think A,
This
because
one?
previous class we talked about how glucose can be coupled with sodium.
Okay, so there's the sodium glucose co-transporter.
Yes, transporter. And it seems like
Would that be an explanation for how glucose could increase or decrease? Decrease. That would be a good argument for a mechanism for how the glucose could decrease. Because, and here's how it would be: if we decrease the sodium gradient, which is what inhibiting the pump would do, inhibiting the sodium gradient would inhibit the energy available for the sodium glucose co-transporter. This is an important concept. This is the idea. Decrease of energy available for the sodium glucose co-transporter, decrease of glucose import or transport inward, decreased glucose concentration inside. Right? Does that make sense? That's applying a principle to that you know, the sodium glucose co-transporter. Good. That's a plausible argument. Okay, let's make an argument for how glucose could increase. John.
There's more free ATP left in the cell, so the cell doesn't have to use as much of its glucose reserves.
That's right. So inhibiting the sodium potassium ATPase decrease use of ATP, decreased consumption, oxidation of glucose, respiration, oxidation. This is getting at another concept from self-is, oxidation of glucose, which is, we have glucose metabolism, right? We talked about that in great detail in self-is, glycolysis, pyruvate oxidation, citric acid cycle, mitochondrial respiration, production of ATP. The cell has a way of sensing its ATP level, and if the ATP level goes up, its consumption of glucose will go down. Okay, so that's what I'm asking you to think about with regard to this question, and you'll do some of that again in a different setting with the problem set. Questions about this? Applying your knowledge of self-is, applying your knowledge of basic principles of transport to answer a problem. What?
I'm sorry, I misunderstood the sodium and glucose. I thought it go same way.
It does go the same way. Sodium and glucose go the same way. So the sodium gradient provides part of the energy for glucose import. If the sodium gradient is decreased because the pump is decreased, then the energy for import is decreased. Less import, less glucose. Both of these are probably going on, and that's why D was the correct answer. It could go either way. And I would, C, you could make an argument for C that they're so perfectly in balance that there was no change. Does that explain? Am I connecting? Am I answering the question?
Bob?
Yeah.
If the sodium concentration inside the cell increase, then
Right.
how could the sodium gradient be?
Okay. Is that the confusion? The concentration of sodium on the inside is low. The concentration of sodium on the outside is high. That was a gradient that was established by the sodium potassium ATPase. If the sodium potassium ATPase this is a gradient. If the sodium potassium ATPase is inhibited, there is less pumping of sodium out. So there is more sodium in. That's the answer A. So if there's more sodium in, then there is a smaller gradient. I don't know how to illustrate that. It's a smaller differential. The differential is the gradient.
That's clear.
Okay.
Therefore, there's less sodium outside of the there's less sodium to travel with the glucose.
There's plenty of sodium outside. There's almost always plenty of sodium outside. You'd have to be really sick to have, well,
Right.
anyway, yes, less gradient, less energy for the glucose input. Yeah, did you say it that way? Yeah, that's the right way to say it. Okay, great. Okay, so now let's turn our attention to signaling. Receptor, neurotransmitters, hormones, receptors, signaling pathways. Those of you who took AMP1 had a full dose of this, and this will be a review. Those of you who are coming here from self-is, that's our starting point. It's important. This section is extremely important for the nervous system. It's somewhat important for all of the physiological systems, especially cardiovascular. So that's why we're reviewing this. So here is a figure from your textbook, which I dislike pretty intensely. So, but this is a cartoon of the GS pathway. It's defined by the receptor that is coupled to the stimulatory G protein, GS. Okay, so this is in your book. It covers some of the material. We'll talk in a minute about why I dislike it so much. We're going to go to a figure from your cell physiology book, which I actually think is more informative and more accurate. And let's fill in the blanks here. So let's work together. I'm just going to start here because I can't reach all the way up. But so what, if we wanted to describe, put into words what the cartoon is illustrating, what might be the first step? And A is just setting up the components. B is the first step of the process. So someone tell me some words that describe what's going on in step B. Diffusion. And binding of the hormone or neurotransmitter to the receptor. Okay, what's the next step? I think we're still in B.
That's trigger a conformational change. in the
Conformational
receptor,
change.
Which attract the alpha, gamma, beta subunits.
Okay, so the conformational change is being illustrated by these little red bars, right? It's different in the unliganded receptor in A and the liganded receptor in B. There's a conformational change in the receptor, which, as Me said, recruits the G protein. In some cases, the G protein is already bound. So conformational change of receptor. Sometimes there is a recruitment of the G protein. That's not always the case. But what definitely happens?
GDP dissociation.
Okay, we'll get there. I'm going to leave a room here for GDP dissociation from GS alpha, the alpha subunit of the G protein. What goes in this room space right here? Conformational change of the GS alpha, the alpha subunit of the G protein. Okay, GDP dissociation. Okay, so now we're moving into here. This is GDP dissociation. What happens then? GTP binds. GTP binds to GS alpha because the site is now available and it's got a high affinity for GTP. So GS alpha, you could call it GS alpha dot GTP now, so it's bound to GTP, or you could just say GS alpha star to indicate that it's the activated conformation of the GS alpha. Then what happens? Separation. That's a good word for what happens. Separation of alpha S star from beta gamma. And that pretty much covers what we now see in this figure. And the alpha S, the GTP bound alpha S, is active. The free beta gamma has some activity too. That's what's this red broadcast thing. Both subunits can have activity.
Does the beta gamma subunit also undergo conformational change as it comes apart from the
Not
alpha
so much.
subunit? Okay.
Not so much. The short answer is the liberation of the free beta gamma activates it all by itself.
Okay.
So it's the alpha subunit that undergoes conformational change and activates it and liberates a now free beta gamma, which is active. And as soon as, and then well, let's go on. Okay, so on the PowerPoint are the answers that I drew, but we covered everything. words on that screen, that word on the board before I erased it? I don't think so. Okay, so now let's think about what happens next. Before we go on, we don't have a cartoon, but let's think about what happens next. I forget what number I was up to. Let's just say 5. I had done a 4, but
Yeah.
now we can talk about step 5. What happens next? Okay, so the question is, are we talking about going back or are we talking about going forward? Let's talk about going forward first. That's a very important question. Are we talking about turning it off or are we talking about continuing the on pathway? Let's talk about the on pathway some more. What happens next?
Secondary messenger.
Okay, we're going to get to second messenger, but you can see I'm leaving a. What does the GTP bound alpha subunit in the GS pathway canonically activate? Adenylyl cyclase. So alpha S star activates adenylyl cyclase. Adenylyl cyclase, what does it do? Adenylyl cyclase catalyzes the conversion of ATP to cyclic AMP. This is a second messenger. In the canonical pathway, that cyclic AMP does what? Anybody remember this from South-Is? Cyclic, I heard a word, cyclic AMP activates PKA, protein kinase A, the cyclic AMP dependent protein kinase, which phosphorylates. Lots of things. Promiscuous. Okay, let's talk about the off pathway. So Zoe's question is a good one. Okay, so how does this system turn off? GTP dissociates is not correct. So the alpha subunit hydrolyzes its GTP. And what is the product of that hydrolysis of GTP? GDP. So now we're in the GDP ligand state. Alpha S GDP is in the off state, and what does it do when it encounters a beta gamma? Binds it, recombines. Thereby terminating whatever signaling the beta gamma might have been done, because it's now occupied by the alpha and not free, not liberated anymore. This should all ring a bell. And if not, you're going to have to review it. And your textbook will help you review it because this cartoon covers it. Receptor, G protein, alpha beta gamma separation, adenylyl cyclase, cyclic AMP, PKA, phosphorylate. It's all here. Let me just be clear. What don't I like about this figure? Well, one is it makes the G protein look very small relative to the receptor, which is not true. The G protein is about the same size. This image is much closer to accurate. See how the G protein complex is on the same scale of size as the receptor, in fact bigger. This protein in this figure makes it look like the G protein is tiny. But it's accurate in some ways. It shows that the alpha and beta gamma subunits are associated with the membrane, and that's correct. whereas this image shows that the alpha and beta gamma subunits are associated with the membrane through a lipid link, which is more accurate. This figure doesn't show anything about GTP binding, GDP dissociating and GTP binding, so it's missing a key step. But it shows that an activated alpha activates adenylyl cyclase, and it shows that adenylyl cyclase is a membrane protein, which is true, and it shows that adenylyl cyclase catalyzes an enzymatic reaction, and it shows that pKa is a cytosolic protein, not a membrane protein, which is true. So there are some things here that are valuable, but it looks wrong. Okay, but there's key information here. So I don't want to tell you ignore it. I want to just tell you, well, look at it with a slightly more advanced eye than you ordinarily might. You'll use this figure for your problem set. That's up. So there's another off pathway. So we talked about the system for turning off of the G protein. But there's another mechanism for turning off of the second messenger. So here's adenylyl cyclase converting ATP to cyclic AMP with some chemical structures for those of you who are chemists. There is in the cell another enzyme called phosphodiesterase, which is important because it hydrolyzes cyclic AMP back to AMP, and AMP is not the same as cyclic AMP, and AMP is not a second messenger, not a second messenger in this canonical standard pathway. So adenylyl cyclase is activated by the active alpha subunit of the G protein. Phosphodiesterase is sometimes controlled and sometimes constitutively active. So when the stimulus is turned on and lots of cyclic AMP is made, cyclic AMP concentrations go up because the cyclase can work faster than the phosphodiesterase. But if this stimulus goes away, the phosphodiesterase will eventually bring the cyclic AMP concentration back down. So be aware of constitutive enzymes, that is to say enzymes that are constantly at work, that can turn on or that can either be part of an activating pathway or a turning off pathway. Okay, so let's work with this for just another clicker. Now, this is just asking you to apply your knowledge. Adenosine receptors, they're called A1Rs, A1 receptors. Adenosine is the neurotransmitter in this case. And actually this is important. This is important in control of the heart rate. We'll talk about that in a week or so. Adenosine receptors are coupled to GI, a different G protein, a G protein that inhibits, does not stimulate, adenylyl cyclase. We mention that in cell phys. We don't talk about GI as much, but it's important. Okay? Adenosine receptors are coupled to GI, and GI alpha, when activated in the GTP ligand state, inhibits adenylyl cyclase. Caffeine, a drug that most of us are familiar with, is an antagonist of the adenosine receptors, and it is an inhibitor of phosphodiesterase. It's a drug that has two biological mechanisms. Okay, so what does caffeine do to intracellular cyclic AMP levels? I'll ask you can work with each other on that to see if you can come up with let's come up with a consensus answer. No. Work with each other, talk this one over, see if you can come up with an answer to this one. I'll be right back.
I mean, that phosphodiester.
Okay, show me your answer. I'll give you a hint, there's one best answer. Okay, I'm seeing a lot of correct answers. Somebody, A is the correct answer. So somebody explain why A is the best answer. Natalie.
So if you're inhibiting the A1Rs, allow the GI receptor to inhibit adenylyl cyclase. So if you inhibit the inhibitor, then you're allowing adenylyl cyclase to work. And then
There's a key concept. Inhibiting an inhibitor allows the whatever else might stimulate adenylyl cyclase to have more power.
And then the same thing with the PDE. The PDE inhibits or converts CAM to AMP, so inhibiting that inhibits
Phosphodiesterase is a turning off thing. Inhibiting the turnoff allows the cyclic AMP to accumulate more. So both of these act to increase cyclic AMP. And this is what happens in your brain and your heart when you drink coffee. You probably have experienced, the energetic effects of coffee, the heart rate effects of coffee. This is what's going on. We'll talk about more. We won't talk about brain effects too much because it's actually really complicated, but we will talk about the effects of cyclic AMP on heart rate and heart contractility, strength of heart rate, real soon. Okay, here's another pathway. So we've talked about GS in detail. We've talked about GI very cursorily, very briefly. And then there's the GQ pathway. This is a figure from your textbook that I've embellished a little bit. So your textbook draws this one. I add a component to this. So let's walk through this. Well, actually, I'm just going to put this up there. It's homework problem number 1. So I'm going to ask you to do homework problem number 1 using this figure as a guide and asking me questions as you're working on homework problem number 1 rather than me lecturing. If I go through this, I will give you the answers to problem number 1. That's something I'm going to resist doing. So I'm not going to go through this one the way I went through GS. I'm going to let you go through it the way in order to answer question number 1. So question number 1 is it's a key concept. Understanding GQ is critical for the cardiovascular system because smooth muscle control of vasculature, the vascular part of the cardiovascular system. So this is important. You'll work with it in your homework. We can talk about it next week if there are questions. The thing that your book doesn't go into is that calcium, in addition to co-activating PKC, I'm giving you a little bit of the answer here, but calcium, in addition to co-activating PKC, also activates an important calcium sensor called calmodulin. And calcium-activating calmodulin activates many enzymes and kinases in the cell. Okay, so I'm adding this component, putting in a little detail in this very vague cell response point. Okay. Zoe, did you have a question? What? Agonist. An agonist is a neurotransmitter, hormone, or drug that activates a receptor. And an antagonist is a neurotransmitter, drug, or hormone that inhibits a receptor. And I'm happy to answer questions about that now, in class, in the hallway if you see me, by a quick email. I'm happy, on the problem set, if you have a question about a question, like, what are you asking here? I'm happy to try to clarify that. But if you email me at 10 p.m. on Sunday, you won't get an answer. So don't wait till the last minute. All right. a key concept that we did touch on in self is, and in AMP1, is this idea of signal amplification. So, and it's, I don't want to go into the, the quantitative nature of this, but the point is, one receptor can generate many second messenger molecules. Each of those second messenger molecules can activate a protein kinase, and the protein kinase, each protein kinase molecule, when activated, can phosphorylate, activate many enzymes, and those enzymes can activate many other enzymes. There's a cascade going on here, and so we have multiplication. If one receptor activated can make 100 molecules of cyclic AMP, and 100 molecules of the kinase can make 10,000, this is another 100-fold amplification, 10,000 molecules, active molecules of the enzyme, and here's another 100-fold amplification. 3 steps of 100-fold amplification, and you get a 1,000,000-fold amplification. So one receptor can be very powerful. And that's the main point, is these effects can be small, but they can also be very large. Okay, so let's do a 2-minute write, and then we'll take a break. So tell me, what are the properties of a so-called second messenger? And then list some of the ones that we've talked about in class today, and others that you may remember from either AP1 or others. So let's go ahead and you can work together on this and come up with a list, and then we'll talk about it, and then we'll take a break.
Wait, did you say yeah? Oh, I thought you said I think. Oh. I'm pretty sure you think because that was activate THC. So I think yeah. Something that is activated here. Scientific word. Something that's activated by something that later activates something else.
This question is 2 of 7 5 points. That's true. Exactly. Most of the wrong
Okay, I just submitted the work order to try to fix the air conditioning in this room.
Great.
Okay, so what are some second messengers? Or no, what are some properties of the second messenger? Well, what are some second messengers? Let's just list some that you know, and we'll see if we can, by identifying them, identify some of the properties. Second messenger. Cyclic AMP. IP3. Diacylglycerol. Diethylglycerol. One more. GTP doesn't count. It's not really a second messenger. Calcium. calcium counts as a second ester. Okay, so what do these have in common? There's, they're in the extracellular? Yeah, so they're cytosolic. They're in the cytoplasm.
Diffusible?
They're diffusible. That's key. Actually, ID3 is diffusible in the cytosol. Diacylglycerol is diffusible in the membranes. And so it's either cytosolic or membrane, but they are all diffusible. They're all small molecules, so not proteins, small molecules. So here's some of the answers that I put up on my PowerPoint. You can fill it in on your handout. Do I give you this one to fill in? Yeah. Questions? Let's take a short break. Go outside and cool off for a minute. Oh, cyclic GMP. We haven't talked about cyclic GMP, but it is a sex ester. We probably won't talk too much about cyclic G. Cyclic A is canonical. Cyclic GMP is another second ester. But we talked about ID3, diacylglycerol, and calcium. Okay, so let's take a short break. Cool off.
I'm like, it's not that great. It was baby's first attempt at digital drawing, but I think
Okay,
it's
so
pretty okay for my first attempt.
I'm going to skip this one just for the interest of time because I feel like I'm running a little slow. So this is a problem that's available to you on Moodle that you can use to check your knowledge. So and if you have, it's not part of the problem set. I won't collect it, but you should be able to say which of these would cause an increase in cytoplasmic calcium and which would not. Go ahead and do that as homework to test your knowledge, and I'll be happy to answer questions about it. But I would like to move ahead, so we'll move very quickly through the answers. And so that we can start to talk about ion channels. This is important, and not just for the nervous system, but it's critically important for the cardiovascular system as well, because the heart, being a muscle, does fire action potentials, and those action potentials, those cardiac action potentials, are essential for controlling the rate and the strength of the heartbeat. So we need to refresh our memory about electrophysiology and ion channels and action potentials. And I'm going to go through it pretty fast because we did this in self-phys. So and this is a nerve action potential. So I do want you to learn about the properties of the channels that are responsible for the nerve action potential, and then we will apply this knowledge to understanding the cardiac action potential, because the cardiac action potential has some similarities and some very important differences that are important to know. But the starting point is the nerve action potential. So the idea is that a stimulus, the stimulus being a small depolarization that reaches threshold, using all the words that you should be thinking about. If this reaches threshold, it opens voltage-gated sodium channels. Sodium flows in. The influx of sodium causes a further depolarization because sodium, the influx of a cation makes the inside less negative. That's how it is. More sodium channels open, more sodium influx, more depolarization, more sodium channels open, more sodium influx, more depolarization, until a maximum activation of voltage-gated sodium channels and a maximum influx of sodium. So the voltage-gated sodium channels are responsible for this rapid upstroke of the action potential. I've skipped a lot of things because we went through these in self-phys. Why is the resting membrane potential negative? You'll have to look that up. The resting membrane potential of these cells, of heart cells, is very negative. We'll be talking about that some more when we get to heart cells. The resting membrane potential is negative. A small depolarization activates sodium channels, which causes a large depolarization. That's the action potential. And then the turning off, two parts of the turning off. One is the inactivation of the voltage-gated sodium channels, and the other is the activation of voltage-gated potassium channels. And that causes a repolarization, or in some cases, a hyperpolarization. Okay, so I'm going to ask you to review your material from cell physiology to understand the nerve action potential. Like I said, because we need to build on that to understand the cardiac action potential. One way that I like to describe this is this so-called Hodgkin's cycle. So let's just go through this. A small depolarization, like from a neighboring cell or from a neurotransmitter receptor channel. Small depolarization, I'm just doing the same story again in a different way. Voltage-gated sodium channels are opened by that depolarization. Sodium flows in, that contributes to the depolarization, which opens more voltage-gated sodium channels. And this positive feedback cycle, very fast, triggers a large depolarization, which is the action potential. I know that I'm using the language and I'm going very fast and I'm asking you to remember stuff that you may have had 2 or 3 years ago, 1 or 2 years ago, usually. But that's your job, and I'm happy to answer questions. Any questions about this? Okay, let's go on. So this is the Hodgkin cycle that describes the rapid upstroke, or the rapid large depolarization of the upstroke of the action potential. And then we have to stop it. Voltage-gated sodium channels, after they open, but with a slower kinetics, go into an inactivated state. So that breaks the cycle. And so the thing that is causing the depolarization is now being turned off. And the depolarization, in addition to opening voltage-gated sodium channels, also, with the slowest kinetics, opens voltage-gated potassium channels. Potassium flows out, and that accelerates the repolarization. So the two things, inactivation of the sodium channels and activation of voltage-gated potassium channels, are essential for a fast repolarization. So now if I go back, you can see what's going on in this slide. The Hodgkin cycle, the fast depolarization, inactivation of sodium channels, and activation of potassium channels, making the repolarization fast. We're going to use both of those for the cardiac action potential, a fast depolarization and a fast repolarization. Fast activation of voltage-gated sodium channels and a delayed activation of voltage-gated potassium channels. Okay? Muscle fires action potentials. The surface membrane of muscle fires action potentials, and that triggers contraction. So now I'm trying to introduce, or trying to remind you of some of the principles of skeletal muscle physiology that you learned in cell phys and AP, because we're going to apply those principles to understanding the heart as a muscle. So again, this is review and a reminder. Your job is to flesh this out, solidify your understanding. So you'll recall from cell phys that muscle contains sarcomeres that contain overlapping filaments of thin filament and thick filament. The thin filaments are actin. The thick filaments are myosin. Myosin has an ATP generated motor. Go through that. But the point here is that cardiac muscle has the same organization of its sarcomeres. I will not ask you to memorize the names of the bands for this because that's detail for AMP1. Okay? So I won't ask you to memorize which is the M line and which is the Z disc. Just know that cardiac muscle has the same structure, sort of the same structure as skeletal muscle. This is critical for how the heart contracts muscle. So just a reminder, the thin filaments are the thin filaments are actin. Actin polymerized into a filament, and then wrapped with other molecules, other proteins called tropomyosin and troponin. So we're going to use all three of these molecules to understand contraction in just a minute. Thin filaments. Actin, tropomyosin, and troponin. Thick filaments are bundles of myosin. Organized around the M line, the heads of the myosin are reaching out in both directions. And here is an electron micrograph that shows the heads of myosin coming off of the thick filament making contacts with the actin. So the heads of myosin make cross bridges with the thin filament, of the actin of thin filaments. So let's go through the mechanism of muscle contraction. The thing that triggers muscle contraction is calcium, a rise in intracellular cytoplasmic calcium. Does anybody remember where that calcium comes from in skeletal muscle? The sarcoplasmic reticulum. Release of calcium from the SR is the trigger for calcium relief, for calcium that stimulates contraction of skeletal muscle. In the heart, we're going to add a little twist to that. That is important. We're not going to make that wrong, but we're going to add something to that. Okay? So, but the key thing here is that in both skeletal muscle and in cardiac muscle, the thing that triggers contraction is a rise in cytosolic calcium. Okay, that's key. So, let's get oriented in this cartoon. Here is the Z disc or Z line. Here is a thin filament attached to the Z disc. Here's a bundle of myosin in the thick filament, and here's one myosin head, one of millions of myosin head in this collection, in this muscle cell. When the muscle is relaxed, when the heart is not beating, when the muscle is relaxed, skeletal muscle or cardiac muscle, the myosin head cannot interact with the actin thin filament because troponin and tropomyosin interfere with that interaction. So the idea here is that troponin, the pink dots, and tropomyosin, the blue rope, prevent the myosin head from interacting with the actin thin filament. That's the relaxed state. Calcium rises when the muscle is stimulated to contract. That calcium, what is the calcium sensor in this story? Troponin. Calcium binds to troponin, causes what are Bob's two favorite words? Conformational change. Calcium stimulated, calcium binds to troponin, conformational change of the troponin, conformational change of the troponin tropomyosin complex moves it out of the way so that myosin can interact with actin. When myosin interacts with actin, it is now in a different, conformational change of the myosin. Okay? The myosin in the relaxed state, and the state that we're still in here, has ADP and phosphate bound to it. Let's just start there. It's a cycle, so you'll see where that ADP comes from in just a moment. The ADP and phosphate are bound to it. When the ADP and phosphate bound head interact with actin, there is a conformational change. ADP and phosphate dissociate. That dissociation causes a further conformational change, and this is the power stroke. The head goes from an extended configuration to a flexed configuration. And that's the power stroke. And that's the power stroke because the myosin is attached to the actin, and it has now moved the actin, pulling the Z disc closer to the other Z disc on the other side, making the muscle get shorter. There's the power stroke right there, triggered by ADP and phosphate dissociation. Okay, let's finish the cycle and then see where we go, how we get. So now we have a configuration that is devoid of nucleotide, right? The ADP and phosphate have dissociated. There is an empty nucleotide binding site. If the cell is energetically viable, it has a lot of ATP. Your cells have a lot of ATP floating around in them if they're alive. The ATP binds to the nucleotide binding site, causes ATP binds, causing a conformational change that causes the myosin to have a low affinity for the actin, and so it dissociates. So it unbinds. So ATP binding causes the myosin head group to detach from the actin, thin filament. Myosin is an ATP ACE. So myosin, this ATP now gets hydrolyzed into ADP. That hydrolysis resets the head group from the flex configuration to the extended configuration, where as long as calcium is high, it could now attach to a different binding site. So instead of binding here, this Z line has moved closer, and so it would attach here, a little further on. And you do that a few million times, and the Z discs get pulled closer to each other, and the muscle contracts.
Can you explain that last little bit again? Like
This
where
last
the
little bit.
the Z line gets closer?
Right, so originally, let's just see. So let's start here. The myosin attached to this spot right here. And it pulled that spot this way. Now, the head group detaches from the actin, hydrolyzes the ATP, and it's no longer going to bind to the same spot because it's now closest to this spot. And so when it does it again, and again, and again, and again, it is pulling on the thin filament and pulling the Z discs, pulling this Z disc this way, and on the other side, pulling the Z disc that isn't shown, which you have to imagine this way, and the two Z discs get closer together and the muscle contracts. Did that answer your question?
Yeah.
So remember, it's a cycle, and the cycle will continue as long as calcium is high and as long as there's ATP. If there is no ATP, then, after death, right? Low ATP, oxidative, oxidation. You're not breathing. The cellular metabolism is shut off. ATP production is shut off because the mitochondria no longer are getting oxygen. Why do we need oxygen? Go back to self-phys. Why do mitochondria need oxygen? Oxidative phosphorylation. It's the final electron receiving. It's the electron acceptor in the electron transport chain, which we need to make ATP. So in the absence of oxygen and the absence of production of ATP, ATP is no longer available to detach the head group from the thin filament. So if the ATP can't help it detach, it stays attached. And that's why muscle gets stiff in rigor mortis. So I went through that really fast. I went through that faster than I may ever have done before. Are there any that I want? So we have nine minutes or so. Questions. We have time to address questions. I went through the action potential very fast. I went through muscle contraction very fast. You're going to need these for understanding cardiac and cardiovascular physiology. So I want to be able to answer your questions now or on Thursday or in lab on Thursday. Okay, well then let's get out of this hot room. And like I said, I'll be available to answer questions between now and Thursday, and we'll do class on Thursday and outside.